Hi friends, here in this video lecture we will discuss about blood clotting or blood coagulation process. How during any kind of injuries there will be clotting of blood and how this will helpful to reduce our losses, that is for blood loss from our body. As we discussed earlier, blood is the liquid connective tissue and this is the responsible connective tissue or tissue we can say that is helpful for circulation of all kind of substances, all useful substances as well as excretory material, that is harmful substances, are circulated from one region to another region in our body with the help of blood. So here how blood clotting occurs and what is the process. So simply here blood clotting or blood coagulation is the process by which the liquid blood is getting converted into a gel state structure. Here this is solidification of blood. This occurs either outside the body at the time of injury or maybe also inside at the time of injury. If we discuss about the duration, how much time duration is needed for clotting of blood, so this is 2 to 8 minute time. At the time of injury there will be initiation of some process and these processes minimum requires 2 minutes and maximum 8 minutes. Even still after that there is no blood clotting, it means blood flow will continue, blood loss will continue, and this is a condition that is known as hemophilia. So the particular person is suffering with a disorder that is sex X-linked recessive disorder, and here in this disorder there is no blood clotting during injury. So there will be continuous blood loss if there is no primary treatment. So how the process is going on? The process or the theory, this was given by McFarlane. Here this is the name of scientist and the theory is known as cascade theory. Here cascade means just gradual or sequential events. So this process completed in 3 steps, but for completion of these 3 steps there is need of nearly 13 factors. So cascade theory proposed by McFarlane for blood clotting, that is a natural defensive mechanism. Here there is need of 13 factors. So these 13 factors are just labeled as Roman numericals. Here 1 to 13 and these are synonyms. So here we are going to memorize these 13 factors with the help of acronym, that is very helpful for you. Now here one more thing that is very important. So when you see here like 1 2 3 4 5 6, here 6th number is missing. So after 5 this is 7, 8, 9, 10, 11, 12 and 13. So factor number 6 is missing. This is given here. This is, or there is no scientific proof for factor number 6. That's why this also known as hypothetical factor. Now about remaining 12 factors. Here this is factor number 1, that is fibrinogen. Before this, what is the acronym? So this is given here. Freshers party today, come on let's sing and call seniors, please have fun. This is the acronym, and here from the acronym we have to take the initials, the initial letter. So from freshers this is fibrinogen, that is first blood clotting factors out of the 13. Now second, this is party, so P for prothrombin. Third, this is today, so T for thromboplastin. Fourth, this is come on, so here C for calcium. Now fifth, this is let's, so L for labile factor. Seventh, as we discussed, sixth is missing. Seventh, sing, so this is stable factor. Eighth, and, so this is AHFA, that is anti-hemophilic factor A. Ninth, call, this is Christmas factor, this also known as AHFB, anti-hemophilic factor B. Already we discussed here A. Now this is 10th, that is about seniors, so S for steward power factor. 11th, please, so P for PTA as well as AHFC, so PTA factor and anti-hemophilic factor C. Now 12 have, so this is Hagman factor, and 13, this is fibrinogen stabilizing factor. So these are 13 factors, all are very important, but out of these here there are very important few. So what are the very important? So first four, this is fibrinogen, prothrombin, thromboplastin and calcium. These four are very important. Now after this, here 8, 9, 10 and 11, these four are also very important and also easy to memorize. This is factor number 8, AHFA, anti-hemophilic factor A. Factor number 9, AHFB, that also known as Christmas factor. Factor number 10, this is steward power factor. Factor number 11, this is AHFC or PTA. This is PTA factor. So out of these total 13 factors, here first 4 factors, next factor number 8, 9, 10 and 11, and 6th number factor is missing. So all these factors are very important. So we are going to discuss further the process of blood clotting that completed in 3 steps, but for completion of that 3 steps there is need of these 13 factors. Now about the steps of blood clotting. As we discussed, there are 3 steps. So here one by one all the 3 are given. Step 1, this is about synthesis of prothrombinase or thrombokinase. Step 2 is about thrombin, and step 3 is about synthesis of fibrin. So step 1, here at the site of injury anywhere in our body there will be some injured tissue. By these injured tissues or cells there will be secretion of thromboplastin. This is an exogenous factor or external factor, while by blood platelets, that also known as thrombocytes, there will be secretion of platelets factor 3. This also known as platelets thromboplastin, that is an endogenous factor. Both these factors in presence of calcium ion, they will synthesize prothrombinase or thrombokinase, that also known as anti-heparin. We earlier discussed heparin is an blood anticoagulant that will stop blood clotting in intact blood vessels, but at the time of injuries heparin will become non-functional. Why? Because of synthesis of this prothrombinase or thrombokinase. So this is anti-heparin in nature. In presence of calcium ion, this prothrombin that is present in blood plasma is getting converted into thrombin. So here pro for inactive. So inactive prothrombin is getting converted into thrombin, and this thrombin is functioning as an enzyme and causes conversion of fibrinogen into fibrin. These are again plasma proteins that is present here in case of soluble situation, and this will converted into very thin protein fibrous that is known as fibrin. These fibrins start to deposit at the site of injury. They will react with blood cells and they will form blood clot or blood coagulum. So as we discussed, within 2 to 8 minutes there will be clotting, and after clotting there will still secretion of a fluid that is known as serum. So this is plasma minus blood clotting protein, or you can say plasma minus fibrinogen. This is serum, and this serum will also stop oozing out after complete deposition of fibrin at the site of injury. So this is all about blood clotting process. Earlier what we discussed about, after these 13 factors there is also need of additional substances for blood clotting. So this is about vitamin K that is needed for synthesis of prothrombin, and also here calcium ion is externally needed. Without calcium ion, blood clotting is not going to be completed. So here, along with 13 factors, there is need of also vitamin K as well as calcium ions. So thanks for this lecture. If you like this, then please share, subscribe and comment. Again, thanks for this.